This is our joint project. I've been working with a Dutch teacher, Andrea, for the last, the guts of 2 years now. Our kind of umbrella is biotechnology. Our aim has been, we're about trying to get them to really get lab skill development going, so they're learning all the fundamentals while learning more about the biology. So using it as a vehicle for them to really get to grips with the fundamentals, right? We start with them with introducing them to biotechnology by learning the basics of microbiology. So they learn about how to grow bacteria, how to make sterile medium, how to use aseptic technique, how to successfully grow these bacteria. We've got lactobacillus growing here, we've got some yeast growing here. The lovely thing about this medium is that it's a selective medium, so it's very forgiving. While they're learning, they're not going to be suffering too much from contamination problems. Give them a problem, give them a question. They've got to figure out how many bacteria are there in a probiotic yogurt, so-called probiotic yogurt. And so they come up with different techniques themselves. It's guided inquiry, so we're trying to steer them into learning specific techniques that they're going to need later on when we bring them to the more advanced kind of DNA technology. So they learn how to use colony count methods. They're sort of standard techniques. There's a lot of mathematics involved along the way. They do serial dilutions, then they plate out the dilutions, and then they're able to estimate from whatever sort of countable number of colonies there are on a plate, they can estimate back to the original sample and estimate, and sure enough there are about 10 billion cells in an average little pot of yogurt, which they didn't think there would be, but it turns out that that's exactly what they find out, right? So anyway, they use the techniques, things like micropipetting and working with really small volumes. They're able to use those techniques to get onto more advanced things like DNA profiling. These are little agarose gel electrophoresis kits. I'll just pop a gel in there for you. So these are little gels. They learn how to pour these gels themselves. This is a technique where you use gel electrophoresis to separate DNA fragments for DNA profiling. So I give them a story and tell them that I've got friends in the horse racing business and that he's given me some samples from horses because they suspect that one of the horses is a clone. And of course they buy into this. I don't prolong the lie. I do tell them eventually that it's just a story. But that, you know, we need to analyze these samples and compare them to the DNA of a famous racehorse, and so they learn the basics of DNA profiling. It's not just about the technique. They need to kind of really get to grips with DNA structure and the chemistry of DNA if they're going to really understand what agarose gel electrophoresis is all about, and then how DNA profiling works and how it does give you a unique pattern that's specific to an individual, and how it's reliable for things like forensics and paternity testing, those kind of things. So once they've learned these techniques, and you can see we're kind of building skills and knowledge as we go, right? Then we've introduced them to PCR. So I was lucky enough to go to Holland in between lockdowns and work with these Dutch students in Utrecht with my partner Andrea. We're doing PCR here. So I developed last year a PCR based on this bacterium here, E. coli 0157. So this is causing trouble all around Europe. It's a Shigella toxin producing strain of E. coli. It doesn't just cause food poisoning. This thing is, and it's particularly common in countries like ours where we've got a big dairy producing sort of component where it's coming from animal waste. That's largely what the story is. So I developed this PCR protocol and looked through the literature to find out what kind of genes are associated with these bacteria. Turns out there's two. There's a Shigella toxin gene. There's a couple of different variants of the Shigella toxin gene. There's an intamin gene, which is an adhesin gene, a virulence factor. I pulled down all the sequences. It's all freely available now. You can get all this stuff off databases, lined up the sequences, and I put together a bunch of degenerate primers. Basically, the idea is it's a mixture of PCR primers so that any strain of these E. coli's, because these things are constantly changing, it's their mutations, right? Any strain of E. coli should be detectable by this PCR protocol. So it's authentic. I give them a bunch of water samples. I have put E. coli DNA into one of them. It's only DNA, so it's perfectly safe. And I spike one of the samples, like I said, A, B, C, D, E, and a bit like the horse racing. I tell them a story that there's an outbreak in a particular area in Ireland. These are 5 water treatment plants that are possible sources of the outbreak. You guys have got to test these samples. Tell me which one it is that is the source of the contamination. So they do the PCR, they come back to their gel electrophoresis, they analyze the results on the gels, and they find where the outbreak is from. And then the last part, my partner Andrea she's a chemistry teacher with a biochemistry, biotechnology background. So I have some bioinformatics activities as well that are to do with E. coli. She's taken it, now this is really nice space for sort of students at senior cycle who are doing biology and chemistry, and bringing it together where they need to have a good knowledge of chemistry, but they also need to have some understanding of protein structure and all those things. So they learn how to do mass spectrum of a protein. Again, it's all online, it's all freely available. We've got all these tools available to our students. We just need to kind of spend a bit of time as biology and chemistry teachers getting to grips with them ourselves, but it's so rewarding because it's so powerful. You know, all the information is there. It's the information age, and it applies to biology as much as it applies to other subjects. So she's got a whole series of activities, which, you know, depending on the level of students that you have, like all teachers, we take stuff from other teachers and we adapt it to our circumstances, to ourselves and to our students. So she's got a whole suite of stuff, all, you know, using online tools and files of mass spectra analysis files that she applies to these activities, showing that you can use this kind of approach as well to get a fingerprint of a protein, detecting Shiga toxin in food samples, an alternative to the way that we use the PCR. So that's it. It's a full suite. It's all kind of microbiology based, all biotechnology, going from really simple to very kind of advanced, all secondary school level. Should be something for everybody.